Now, when it comes to non-disjunction could happen with our autosomes, or it could happen with our sex chromosomes, okay? Non-disjunction does not discriminate. It could happen with any pair of chromosomes. For this specific lab, we are going to focus on non-disjunction in regards to your sex chromosomes. There are several disorders that we're going to talk about in regards to non-disjunction with your sex chromosomes. So that's what we're going to focus on. So we're really, again, from here forward, we're assuming that the autosomes separate fine, okay, that the gametes that we produce all have 22 autosomes. We're just focused on the sex chromosomes and what happens if non-disjunction happens with our sex chromosomes. In other words, if they don't separate properly. So to begin, I want to talk a little bit about developing your gametes. So spermatogenesis is the production of sperm via meiosis in the testes of male. And spermatogenesis is going to start with a diploid cell called a spermatogonium. And the spermatogonium is going to ultimately divide, and it's going to lead to the production of what we refer to as sperm. So again, notice in my little picture here, I'm not showing you the autosomes. I'm only showing you the sex chromosomes. Please know that the autosomes, we're assuming that they're all here and that they're dividing the way that they should. Non-disjunction is not affecting the autosomes at all. So again, in normal spermatogenesis, what should happen is during interphase, specifically synthesis, all the chromosomes are going to be duplicated. So here is my X and my Y. Again, spermatogenesis, the sex chromosomes are an X and a Y, denoting the male, right? The SRY genes are on that Y. So we're duplicating those chromosomes. So now we have two X's and two Y's. We should, in meiosis 1, we should divide those chromosomes. We should pull them apart. The Y's should go into one daughter cell. The X's should go into another daughter cell. And then during meiosis 2, when we separate the sister chromatids, we should ultimately end up with one Y in these two daughter cells and one X in each of these daughter cells. So again, what we're assuming is in this cell right here with this Y, you would have 22 autosomes and one sex chromosome. You have the Y, that's a total of 23, right? Same goes for this cell. This cell right here would have 22 autosomes, one sex chromosome, but the sex chromosome here is an X. We still have the normal 23 chromosomes. So let's envision what will happen during fertilization. So here's my little sperm, right? This little sperm right here, it's carrying an X sex chromosome. It's got 22 autosomes. If it fertilizes a normal egg, and when I say normal, I mean it's got 22 autosomes, one sex chromosome, eggs always only have an X. What's going to happen is when it fertilizes, it's going to result in a zygote with 46 chromosomes. Okay, 44 autosomes and two sex chromosomes. The two sex chromosomes would be an X and an X, and you would result in a female. If a little Y-carrying sperm fertilizes the egg, again, it's a normal egg. We have 22 autosomes, one sex chromosome. Here, my sperm has 22 autosomes, one sex chromosome. When fertilization happens here, right, it's going to result in an individual with a normal number of chromosomes, 46. The sex chromosomes are going to be an X and a Y, resulting in a male. So now let's talk about if non-disjunction happens, okay? First, we're going to look at non-disjunction if it happens during meiosis 1, and then we're going to look at if non-disjunction happens during meiosis 2. So again, what we just went through was normal spermatogenesis. That's what we expect to happen, right? But now we have our little spermatogonium. Again, during synthesis, the chromosomes all get duplicated, but unfortunately in meiosis 1, we fail to separate the X and the Y's, and they all go into this daughter cell, whereas this daughter cell right here doesn't receive any sex chromosomes. Still has autosomes, okay? So it still is going to have the normal number of autosomes, but no sex chromosomes. Now when we go and divide the sex chromosomes, this cell right here is going to get an X and a Y, right? So X and Y go one, X and a Y go another. And unfortunately, these two sperm that are produced, they don't have a sex chromosome, Okay, they don't have an X or a Y. So again, if we flip back, this is what we expected, right? You should have a single sex chromosome in each of the gametes. But instead, if non-disjunction happens in meiosis 1, all of my gametes are abnormal. These two gametes have one too many sex chromosomes, and these two gametes here have one too few. They don't have a sex chromosome. So then what does that lead to? So if this little sperm right here, right, if this fertilizes my normal egg, this little sperm right here has 24 chromosomes. 24. How many should it have? Hopefully you're sitting there going, there should be 23. Yeah, you're right. There should be 22 autosomes and one sex chromosome. This has 24 because it has 22 autosomes and it has 2 sex chromosomes. So when this fertilizes my egg, it's going to result in a zygote with 47 chromosomes. Okay, 47 chromosomes. It's going to have my normal 44 autosomes. That's great. But unfortunately, it has 3 sex chromosomes instead of 2. The sex chromosomes that it's going to have, the egg will give an X, and the sperm is carrying an X and a Y. So the sex chromosomes that this individual will have are XXY. Now remember, this individual is going to have 47 chromosomes in all the cells that make up their body, because once these fuse together and form that zygote, then mitosis begins, and it divides and divides and divides until it forms that fetus. So in this case, this condition, Klinefelter syndrome, if you end up with someone that has an XXY, it results in a condition called Klinefelter syndrome. That results in a male with feminine features. Sometimes, or oftentimes, they may they may have some learning disabilities. They may not test on standardized tests as high. It can also lead to some feminized features, so they may have larger breast development, for example. But these are all sort of signs of the Klinefelter syndrome. Now, the way to diagnose this, right, would be to take cells, get the DNA, and to do a karyotype in order to figure that out, in order to actually see what the chromosome number is in the cells. If this sperm, right, so this sperm has 22 autosomes, no sex chromosomes, right? So here it is, 22 autosomes, no sex chromosomes. If this fertilizes my egg, what this is going to result in is a zygote that has 45 chromosomes, not 46. So again, 46 is sort of quote unquote what we expect. 23 from dad, 23 from mom, 46 in the zygote. But non-disjunction happened. This sperm is fertilizing this egg. It results in an individual, in a zygote, and eventually in a fetus with all of its cells, right, having 45 chromosomes, 22 autosomes, 22 pairs of autosomes, and ultimately only one sex chromosome. It has an X. Turner's syndrome results in a sterile female, and she tends to be below average in height. Now, again, to diagnose this, one of the ways that you could do this is by taking a karyotype, all right? Or doing a karyotype, I should say. That would be a very quick way to really figure out whether or not someone has a genetic condition based on having an extra chromosome or missing a chromosome. Now, one thing that I do want to point out when it comes to the sex chromosomes, you must have at least one X sex chromosome. If you only have a Y, the individual will not develop. Okay, we refer to that as being non-viable. So it's okay to only have one X, but you have to have at least one X in regards to your sex chromosomes. Okay, now let's look at what happens if non-disjunction happens during meiosis 2. So when non-disjunction happens during meiosis 2, what you're going to see here, right, is here's my spermatogonium. Again, during interphase, the chromosomes are duplicated. Now they split fine in meiosis 1, right? The X's come down here, the Y's come up here. But now we sort of have 3 different scenarios that could happen, okay? Worst case scenario, right, we have non-disjunction in both. So both Y's go into one gamete, no sex chromosomes go into the other. Both X's go into one gamete. No sex chromosomes go in the other. Otherwise, in this case, 100% of the gametes are abnormal in regards to sex chromosome number. Or it's possible that the X's divide fine. So these two gametes here, these would be normal. They would have 23 chromosomes, 22 autosomes, and 1 sex chromosome in X. But notice here that non-disjunction happened. So 50% of the gametes are affected. This gamete would have 1 too many chromosomes. This gamete would have 1 too few chromosomes. Or, right, the Y's could separate properly and the X's could fail to separate. The X's, this cell right here goes through non-disjunction in regards to the X chromosome, and both X's end up here and no sex chromosomes here. Again, in this case, 50% of the gametes are normal. This would have 23 chromosomes, 22 autosomes, 1 sex chromosome, 22 autosomes, 1 sex chromosome. This would be an abnormal gamete. This would have 24 instead of 23, 22 autosomes and 2 sex chromosomes. This gamete would also be abnormal. This would have 22 autosomes, no sex chromosomes. So now let's do what we did before, right? So if a normal sperm, meaning that it has 1 sex chromosome and then it has 22 autosomes fertilized by normal egg, this is my normal egg here, right? 22 autosomes, 1 sex chromosome. What's going to happen is this is going to result in a female with the right number of chromosomes, 46. 22 pairs of autosomes, 1 pair of sex chromosomes, X and X. That's this individual right here. In this case, if my normal egg is fertilized by a quote-unquote normal sperm carrying the right number of chromosomes, it's going to again result in a male that has 44 autosomes and 2 sex chromosomes, 46 total chromosomes. In this case, right, their sex chromosomes are going to be an X and a Y, and it's going to result in my quote-unquote male. Now, we also have another couple of situations that we have to examine, right? Just like before, if this sperm right here that doesn't carry any sex chromosomes fertilizes my egg, right, it's going to result in a condition referred to as Turner syndrome. So 44 autosomes, but only 1 sex chromosome, right? Only the X is what we get here. And as a result of only having that 1 X chromosome, it results in that condition that we refer to as Turner's. There are 2 additional conditions that we want to touch on here. There's this sperm right here that had 2 Ys, and there was this sperm right here that had 2 X's. So you can see those 2 examples here. So if a sperm carrying 22 autosomes and a Y, so again, this sperm would have 24 chromosomes, fertilizes my normal egg, it's going to result in an individual that has the sex chromosomes XYY. And you can see I listed that right here. So XYY is a condition called Jacob syndrome. Individuals with that genetic makeup are generally what we consider sort of normal male. They might be above average in height, and sometimes they have severe acne that accompanies this. The other scenario is if we look at this little gamete here, this little sperm carrying 2 X's. Again, this individual would have 24 chromosomes in the sperm, right? Or I should say not individual, but this gamete would have 24 chromosomes, 22 autosomes, and 2 X chromosomes, 2 sex chromosomes. That makes 24, right? Should only have 23. If that fertilizes my normal egg, again, my normal egg has the 22 autosomes and the 1 sex chromosome. It's going to result in the sex chromosomes in an individual zygote that has the sex chromosomes XXX. Again, this zygote is going to have 47 chromosomes instead of 46, right? 44 autosomes, 3 sex chromosomes, 3 X's. That's a trisomy condition. This condition is called poly-X syndrome, which generally results in a normal functioning female. So as you can see, right, if non-disjunction happens, and we've just now looked at spermatogenesis, if non-disjunction happens in regards to spermatogenesis, we can get a number of different outcomes, right? If it happens during meiosis 1 in spermatogenesis, 100% of the gametes are affected. And if any of these sperm fertilize a normal functional egg, it is going to result in some sort of a condition. If non-disjunction happens in meiosis 2 of spermatogenesis, again, you can get a variation. You may end up with a normal female with the right chromosome number, or a normal male with a correct chromosome number, or you can end up with any of these types of conditions. So one of the things that we need students to be able to do is you have to be able to explain, well, what happens if non-disjunction happens in meiosis 1? What are the gametes that I would expect to form? What happens if non-disjunction happens in meiosis 2? What are the gametes that I expect to form? If these sperm fertilize these eggs, right, what do I expect the chromosome number to be, right? Should it be XXY? Do I expect that, the cell would have 47 versus 45 versus 46? So really demonstrating your understanding of non-disjunction is really what we're after here by really applying it to the sex chromosomes. Now, you as individuals, so that was all sort of done in your lab book for you. What you need to do is now go through and do this with oogenesis. With oogenesis, again, this is what sort of normal oogenesis should look like. In oogenesis, when we form our eggs, in regards to our ovaries, one of the things that happens, the chromosomes are going to duplicate. Again, this happens in synthesis of interphase. So we can see our X and our X. We've now duplicated them. When we divide the chromosomes, one of the things that you'll note is that these cells are not the same size. That's because when we do cytokinesis, there's an unequal distribution of the cytoplasm. So we end up making one larger cell in one smaller polar body. This polar body is not functional, okay? It can't be fertilized and develop into anything. But ultimately, it's this cell right here that we're then interested as it goes in and does meiosis 2. This is going to, again, after the sister chromatids are separated and the cell goes through cytokinesis, we again do an unequal distribution of the cytoplasm, producing sort of what we think of as that functional egg or ova and the other polar body. The reason that the majority of the cytoplasm and organelles end up in that sort of primary oocyte, and why that's important, is if it's fertilized, okay, and the secondary oocyte divides during meiosis 2, it's ultimately going to have to sort of sustain itself until it can sort of be nourished by the body and implants eventually into the uterus. And so literally, eggs are very energetically expensive to make. For every time that a female really does oogenesis, and again, I should back up and tell you that in regards to oogenesis, females are actually born with all of the eggs that they're going to have. So a female baby that's delivered actually has all of her eggs already in her ovaries. Once she hits puberty, she'll begin to release those eggs when she ovulates each month. But for every one time through meiosis, and again, we're not going to get into the details of this. They're much more complex than it might appear than normal meiosis. We'll talk more about this if you do go on to anatomy and physiology. For every one sort of oogonia that goes through this process, females only produce one functional egg. Males, on the other hand, right, if you remember back to when we just did this with males really do a very typical meiosis, right, where they're producing 4 functional gametes. Okay, so again, eggs are energetically more expensive. They have to sort of provide the organelles, they sustain the cell division. The sperm are really a vessel to deliver sperm. And so when males do spermatogenesis, again, they get those 4 functional gametes. Females, only that 1 functional gamete. These polar bodies, again, not going to be fertilized. What we want students to do is now go through and basically fill in the circles in what you expect to happen if non-disjunction happens in meiosis 1 of oogenesis or non-disjunction happens in meiosis 2 of oogenesis. So again, what does that mean? right? In your book, you can see that there's an X and an X drawn for you. You then see this. So if non-disjunction is happening in meiosis 1, the idea is all of these sex chromosomes are getting pulled into this cell, and then they should divide, right? But notice up here, right? There should be only one X. Down here, there are 2. That means that these gametes are all going to have an abnormal number of chromosomes, right? These gametes have 24 instead of only 23. So make sure that you go through and you do draw out non-disjunction in both meiosis 1 and meiosis 2. Also make sure that you're answering all of the questions on pages 153 and 154, the questions that have to do with this non-disjunction and oogenesis, because that will really illustrate whether you understand this or whether you may need some additional help.